Okay, thank you so much. You are welcome. We just want to try out and just make sure that everything is okay from our end before we begin at 17 exactly. Just a few minutes from now, I will invite our team leader, Mr. Helmut Anschutz, to just come and give us a brief introduction about today's presentation, and then we'll be good to start. So maybe before we begin, some bit of ground rules. We are all expected to mute our microphones so that we can have a smooth interaction. Then should you have any concerns, you can use the chat box to just be able to write your concerns or questions, and then we'll be able to address them later on. So I will invite the team leader now to come and give us an introduction for today's presentation.
Yeah, thank you very much. Good afternoon, everybody. I hope you can hear me. We are very unlucky with the weather. First, the rain started some days ago, and we all had a good planting rain, so we are busy planting. So many of you could not take part in our meeting here at AKTC. And now here in Chisamba, we have a heavy rain again. I hope the internet remains stable and you can hear us clearly. The rain makes a lot of noise on that roof. I see, Bruce, you don't have your microphone switched off. Do you read me?
Sorry, there's two Bruces. This is Bruce Skinner, but I can hear you. I'll switch my microphone off again.
Oh, that's fine.
I think that
Bruce.
might be Bruce Dankwitz.
Yeah.
Hi, Helmut. How are you doing?
I'm well, thank you. Thank you. So I hear you. So I go ahead. Yeah, my name is Helmut Anschutz. I'm the team leader of the Zambian German Agricultural Knowledge and Training Center. We are based on Golden Valley Agricultural Research Trust in Chisamba. And yeah, we are very happy that we have managed to get a well-known mycorrhiza expert, Dr. Schneider from Germany, to Zambia to present on her findings. I cut a long story short. Dr. Schneider is founder and CEO of a company called Institut für Pflanzenkultur in Germany that was founded in 1995. And since 2001, she has a second company, the INOC GmbH. And INOC is working with mycorrhiza. So that means since 21 years, there is an experience with mycorrhiza from Dr. Schneider. Her scientific education is she did a PhD at the Institute of Applied Botany at the University of Hamburg. And she was a part-time lecturer at the University of Applied Science and Art in Göttingen. So you see there's a big experience. I told when we had our presentations here at AKTC, I said, just come in with your questions. Dr. Schneider has vast resources. She probably could talk for 8 hours or longer about mycorrhiza and soil life and all that. So she will do a presentation, and please just type your questions into the chat, and then we try to answer them immediately. By saying that, and still 1 minute to go, so I hand over to Dr. Schneider to start with the presentation. And yeah, thank you very much for joining us. The presentation, we will record it so it can be watched at a later stage. You are all welcome, Dr. Schneider.
Yes, thank you for the introduction. let's see if this adventure will work today because of this weather and the connection, but I do my best. I will start with a PowerPoint presentation, and later we will have time for questions, whatever you would like to know. Thanks for the introduction. I will try to share my screen and share the presentation with you. And the very beginning is a short video, but now I cannot see the presentation anymore. Ah, now it's there. Okay, it was too big. So you should see now one slide or two slides. One. This is perfect. So you see the title of my presentation, the use of biostimulating mycorrhizal fungi in agriculture. And I will give you an overview. So next slide will be the video, the short video for introduction. Okay, this is two slides, what you see. Let me change, but I don't know where. There's something like exchange monitors, because it's nicer, of course, for you when you only see one. But I cannot find it here. So, yeah, might be because we have two monitors here. Yeah, something is happening. Okay, now I can see. Now, yeah, it's not very severe. So you concentrate on the bigger screen. I don't know how to change it now. Usually there is a button to exchange the presentations, but I cannot see it here. What I introduced is the video, which is starting in just some seconds. It is made for trees, but you can always, when trees are mentioned, you could exchange that word with plants in general. So let me see if the video is working. Of course, we tried several times.
Trees may look like solitary individuals, but the ground beneath our feet tells a different story. Trees are secretly talking, trading, and waging war on one another. They do this using a network of fungi that grow around and inside their roots. The fungi provide the trees with nutrients, and in return they receive sugars. But scientists have found this connection runs far deeper than first thought. By plugging into the fungal network, trees can share resources with each other. The system has been nicknamed the Wood Wide Web. It's thought that older trees, fondly known as mother trees, use this fungal network to supply shaded seedlings with sugars, giving them a better chance of survival. Those trees that are sick or dying may dump their resources into the network, which might then be used by healthier neighbours. Plants also use fungi to send messages to one another. If they're attacked, they can release chemical signals through their roots, which can warn their neighbours to raise their defences. But like our internet, the Wood Wide Web has its dark side too. Some orchids hack the system to steal resources from nearby trees. And other species, like the black walnut, spread toxic chemicals through the network to sabotage their rivals. Arboreal cybercrime aside, scientists are still debating why plants seem to behave in such an altruistic way. The hidden network creates a thriving community between individuals. When you're next in woodland, you might like to think of trees as part of a big superorganism, chatting and swapping information and food under your feet.
So, okay, that should serve as an introduction. Now, the outline of my talk today of the short seminar is I will say some words about us so you know who is talking to you. I will explain mycorrhiza further. I know that you have different knowledge. For some it is very new, some already have experience, and I will show some success stories. So what was already mentioned, 27 years ago, I started with the first company, and it wouldn't be important for you, but with it, the first company is dealing with plant production. And with this plant production, we came to the mycorrhiza, and that's why I tell it, because we were, let's say, our first customers. We wanted to use it at that time because we had so nice results, but there was no market, no product, we couldn't buy it, there was nothing on the market. So, and as we like to transfer knowledge from science to application, we started that second business, Enoch, which is dealing with commercial mycorrhiza production since then. But in the first 15 years, let's say, not for agriculture, but for all different kinds of plants, for revegetation, for roof greening, for these application fields, agriculture was not open for us, let's say, because we first had to optimize the formulation and the product. So we worked hard together with our academic partners to get that ready. We are located in the north of Germany. And we have everything what we need to do the work. We are working in a beautiful farmhouse from 1880. We have lab facilities, sterile working places, greenhouse, and yeah, whatever we need. And at the moment, we do the work with 30 colleagues, and quite a number are scientists, so have an academic background because we still do a lot of science. Here is a summary of soil life, of what is in beneath our feet, which we tend to underestimate because we cannot see it. Most of the organisms we cannot see, which is composed of bacteria, a lot of bacteria per gram soil, fungi. I don't know how they made it, but somehow somebody calculated that we have 10,000 kilometers of fungal hyphae in a cubic meter of soil. So this is quite a lot. And of course, what we tend to see is the small insects or you know, like earthworms, for example. This is what we see. This is the microfauna. So, and all these organisms are in the soil. They are working together. They are working against each other. So like the video introduced, there is a lot of physiological communication going on in the soil. When we come to mycorrhiza, presentation stopped now. It is already quite old. The term mycorrhiza was introduced in 1880s by two different scientists, a Polish one and a German one. And since that, we use the term mycorrhiza. It is composed of the Greek words for fungus and for root. So it's like fungus root or fungal root. So this is what mycorrhiza means. And so this is it's not easy to write and work with that term, but it's right. It says the right thing. When we look on mycorrhizal associations, there are many, many different fungal species, let's say, in general. And 90% of all plant species are used to working together with fungi. So it's not at all an exception, but it's very normal for plants to have these fungi at their roots, to collaborate with them. And so we have to take care, we as growers, as farmers, we have to take care that our crops are finding their partners in our industrially, let's say, managed farm soils. There are different ways and groups of mycorrhizal association. I will only mention the two most important. Which is this one. This is the smaller group of mycorrhizal fungi. This is ectomycorrhiza. 2% of plants are using that collaboration, and it is mainly woody plants. So in forests, needle trees, they use ectomycorrhiza. And the term already points into that direction. The fungi, which is here shown in blue, they keep outside the plant root. So they grow outside the plant root and only in between the cells, let's say. They don't grow inside the cells. That's why this symbiosis is called ectomycorrhiza. So it stays outside. The next and much more common group is this here on the left side in red again. This is the arbuscular mycorrhizal fungi. The majority of plant has hosts, and this is about 74% of all plants, they host arbuscular mycorrhizal fungi. This is, so it makes our life a bit easier because almost all crops need arbuscular mycorrhizal fungi. There are very few exceptions, let's say. And another term for this arbuscular mycorrhiza is endomycorrhiza. So this is more or less the same. Endo because they do grow inside the plant cells. So they really, and this is seen here on the scheme, they really grow into the plant roots, plant cells, and form their organs in there. At the moment, we know around 300 different of these mycorrhizal species, fungal species, and the specialists guess that we will find up to 1,500, but this is still, nobody knows. And then there are the other groups of mycorrhiza are not that important for crops. That's why I don't mention them here. But show you in this picture the symbiosis, how it looks. For example, this example is a potato plant, well known, you can see it there. And it has not only the tubers, which we eat, but as well normal roots. And these roots are well colonized with mycorrhiza. And on the left side, there is such a root. This is a magnification of that root fragment. The brownish part is the root, and everything which is stained in blue is fungus. So on the first sight, you already see it's quite a lot of fungus inside such a root. And there are different organs of the fungus, so that are mentioned here. The arbuscules, where this is little trees, that's why the name is arbuscules, look like little trees. They are the location of the exchange of nutrients between fungus and plant. In vesicles, the fungus stores the nutrients, and then we have hyphae, where the fungus grows, let's say, and spores, of course. Some fungal species produce spores inside the roots, and some do produce them outside the roots. So this differs. And this is important too, we have plant species which are non-mycorrhizal plants. So if we use them as crops, there's no need to give them mycorrhiza, because they cannot make use of it. It won't harm them, but you can save your money. So there are not too many crops among this group. I gave some examples here. It is sugar beet, cabbage, canola, and mustard, for example. But those are the main crops which we use, which are non-mycorrhizal plants. So they found other ways to get access to nutrients. Here again, a picture of the mycorrhiza inside the root. And what I would like to show here is this little tree-like structures in magnification here. You can, I think everybody can imagine that it is a very good location of exchange because of this, these small structures which make that possible. And on the left side, there is a scheme, is still in German, but easy to understand. The white circle is the root only, let's say, where the root is able to get access in the soil to water and nutrients. And the green circle is the, let's say, wider area where mycorrhizal fungi can help the plants to accumulate water, phosphorus, nitrogen, and in return gets carbohydrates from the plant, from the photosynthesis of the plant. So the plant pays with sugar, let's say. And when this symbiosis is working well, we have all these nice beneficial effects on our plants. They don't necessarily come all at the same time in the same situation, and sometimes they don't come at all because the plant is happy with the situation. So if we manage that our plants are perfectly maintained, they probably won't take service of the mycorrhiza because they have to pay for it. So that can happen that there is no mycorrhizal effect. So it's not a miracle cure, but normal limitations, let's say. What do the fungi do? They can enhance plant growth, plant tolerance, of course, product yield, which we are all very interested in, but as well product quality, and I give some examples on that. Water absorption, which is very important in nowadays because of drought situations we have. Nutrients intake, I think this is the most well understood effect. And then soil amendment, this is personally my favorite or one of my favorites because it is the basic of other beneficial processes in the soil. So if we have a well aggregated soil with crumbs, then all other microorganisms and soil organisms, they are happy too. And I will show that in some slides. Because we are a mycorrhiza producer, I will show, of course, many examples of application and good results. But first, I would like to explain a bit how we do produce those fungi, because that makes a difference. And there are products on the market there are at least two ways to produce it. And there are products on the market of both methods. We do the commercial production like it is shown here on those pictures. So we have large boxes, let's say, we grow plants inside these boxes and we have a substrate. So these partners have to be there. We grow, and the fungus, of course, is introduced into the system in spring. And then we grow the partners together 3 to 4 months. Of course, our interest is not nice corn plants. You see the corn plants here. They don't look very happy. And this is with purpose because we are not interested in this case in corn plants, but in mycorrhizal fungi. So, but the principle is like sourdough. You put a small amount to substrate and then it grows. So quite easy, I would say, but of course, challenges are in the details. And here I show the different possibilities how you can produce it. They are all starting from soil collection. So from soil samples, which I put here in the red circle. When we go first on this kind of blue part, this is a very simple method to produce mycorrhiza on farm. So this is what our ancestors did sometimes. So when they planted, for example, blueberry, new plantations, they went to the forest, digged out some soil and put it to their blueberry plantation. So this is one, let's say, low input, low cost method to produce it on farm. Everybody can do it, but of course it is not at all controlled. So you don't know because you don't have a microscope, you cannot have a look what is going on, if there's enough fungi, if there's the right fungi or fungi at all. So we don't know. That's why it's not a bad method, but we don't use it for commercial production. We do use the green method, which is called ex vitro production. So this is in greenhouses. So open system, and I will show what advantages it has. So this is the system you already saw on the pictures. And then we have another method. This is here called, this is this orange part. And this is called production in vitro. So this is in the lab under sterile conditions. We take spores as well from soil samples. We sieve them out of the soil, isolate them, select them, and disinfect them, because inside this lab method, they have to be very sterile, very clean. And we don't allow other fungi or bacteria running around there, because this will destroy the whole system. And then it is possible to produce mycorrhiza under sterile conditions. we always need at least plant parts. So in that case, we use not complete plants, but root organ cultures. And with these root organ cultures, the fungi are able to grow. Because an important detail here is that we always need, for the production of arbuscular mycorrhiza, we always need plants, or at least plant parts. We are not able, nobody is able to grow them only on culture medium. There always have to be a living plant around. And this might answer already questions which I often hear. I have a very nice compost. Do I already have mycorrhiza propagation inside? And as there are no living plants inside my compost, the answer is no. We can discuss this later. I put here some pros and cons of the two methods, and I don't expect that you read them all, but to give you, let's say, the main aspects is that this in vitro method is very clean. This sounds nice, and it is. For example, we use it as starter in Oculum because it is so clean, but the mycorrhiza is missing other team members, let's say, so-called mycorrhiza helper organisms. So mycorrhiza is working together with other microorganisms too. And then in these in vitro produced products, they are not there. As well, the spores are very weak. They are smaller and less robust than the greenhouse system. And this is another reason why we do all our commercial products via the greenhouse system. So I will leave that topic and come to the ectomycorrhiza. So the smaller group of mycorrhizal fungi, but still it has an importance on the market. These are fungi which we are able to grow without plants. So this makes our life a bit easier. We can grow them on petri dishes, and upscale the production in solid state fermenters, so glass jars or big fermenters, depending on what we need. So this is possible as well. The product then is this kind of powder which is sold. And I would like to introduce as well, this is the last slide towards the quality, because it is important. There are quite a lot of bad products on the market because customers cannot see mycorrhiza. So in Germany, for example, we until this year, we didn't have any registration or legislation for these products. It's a bit astonishing because Germany is well known that everything is regulated. But for biostimulants, which is the correct product group, there were no rules at all. Everybody could put a label on whatever dirt and say it is mycorrhiza. And some companies did, because mycorrhiza is kind of hyped at the moment. So of course, we don't like that because our customers, the products cannot work because there's not enough mycorrhiza inside or not enough living mycorrhiza. So we state that the minimal quality of a mycorrhizal inoculum is the capacity to generate mycorrhiza, because if it doesn't do that, it is whatever, it's a fertilizer, but it is not a mycorrhizal inoculum. Then, and this is multi-factor dependent, of course, we all are interested in positive beneficial effects on plant performance, because this is what we eat or what we sell. But this we cannot, we as a producer, we can, of course, we work, we do do many trials, field trials with different crops and situations, but we cannot cover all possible situations. And what I said in the beginning, when the plant is very happy, it won't take the mycorrhiza. So there are two questions. Does this product contain a sufficient amount of mycorrhizal propagules, and are they viable? And then, of course, it should have a mycorrhizal potential. So it should be able to create mycorrhiza. And yeah, now I will come, and by the way, when I say mycorrhizal propagules, people say as well units or similar terms. It is, and I mentioned that in here below, mycorrhizal propagules are spores, mycorrhizal root fragments, and living hyphae. So it's not only spores which are important. as well the other parts of the fungus. And there are products which only contain spores. They leave out the 2/3 of the fungal potential, which is something. So now I come with all these facts I wanted to explain to you. I come to some explanation what the mycorrhiza does. I start with some statements on water potential. So here I can explain that mycorrhiza keeps water in the soil. And this I can show you with a short video in the next slide, but here let me explain. On the left side, you see sand particles laying around in the soil, or soil particles, and water. This is a typical soil where no mycorrhiza or not enough mycorrhiza is present, and the soil particles are not at all connected. On the right side, you see the mycorrhiza aggregated soil, let's say. So the soil particles are with a kind of glue, they are sticked together, and the water can physically stay inside these particles. So this is a big advantage. We have these capillary pores, and they can keep the water inside these aggregates. Outside the aggregates, there's air in the soil. And this is an advantage too, because the plant, of course, needs both. It needs water and needs air in the soil. So now another experiment with another small video, not working. So, but I think it's important enough. I would like, though, to show it to you, but for that, I have to, let's say if it works here, I have to open another file. Okay. And then I hope it will start. So let me explain you what you will, oops, now I made a mistake. What you will see before I started, it is on the left side, there are two glass jars on the, and in each of the glass jars, there is a plant root system without the pot. The left example has been grown with mycorrhiza fungi, and the right example, the right plant has been grown without the fungus. And when I started, you will see that somebody pours water on those two root bales. And see what happens when the water's, is it well seen, or what do you do? Is it big enough? Yeah, okay. So it's full screen. So what you see is the result here. You see that on the left side, the root bale was able to keep the soil, to stick together. And on the right side, without this glue, it is called glomalin, the soil is not sticking together, and only pouring water like a heavy rainfall destroys this kind of soil. It is only a loose connection of the soil particles. And this, you can easily imagine that this soil is then under the risk of erosion. The heavy rainfall will take away the fine parts of the soil. So I will go back now to my presentation. Let me see. Yeah. You should be able to see it again. And we'll go to the next slide. Because now I give you a number of examples out of agriculture. Of course, they are from Germany, German agricultural fields, German farms, but yeah, there is a lot of knowledge and published knowledge in, maybe you cannot see them all. Let me try to improve that. So now you can see the whole thing. I start with quite a simple graph here. But I can explain many things with that. You see there is mycorrhization in percent. and different mycorrhiza, let's say, factors. One is F% and one is M%. F% is the frequency of the mycorrhiza in the root system. So when I investigate a mycorrhizal root under the microscope, I count every time when I see there is mycorrhiza. So this makes F%. But M% is the intensity of the colonization. So there is a difference if there's only a slight piece of hyphae, mycelium, so there's only small mycorrhiza, but it is, yes, it is mycorrhiza, but not very intensive. So we take those two factors to, let's say, rank samples, soil samples or root samples according to their mycorrhization. And you see here different things. The light gray bars are the control. And you can see that they have already root colonization of around 40% F, which is already quite good. So this is an example, it's more an exception of a farmer who already has quite a lot of mycorrhiza in his roots and in his soil. But this company, the farm, is managed he's doing organic agriculture, but as well conservation agriculture since almost 15 years. So there's no big surprise that this has some effects, beneficial effects on the root organism, on the soil microorganisms. But when we added mycorrhiza, and this is the green bar, we can see that there is a significant increase of the mycorrhization. So we bring it up to 52%. So that would be an example where I can see, okay, the introduction of mycorrhiza into the soil works. There is more mycorrhiza with inoculation, but the 40% would be okay as well. So most samples, what we see, they have 2%, 4%, below 5%. So, and they really have to do something. This farmer can already be quite happy. So let me go to the next. You might ask how to get it into the system. There are different ways. One is to bring it out with a granulate spreader. This is here on the drilling machine, and this is how we did it for a field trial this year in May. We put the mycorrhiza in the small tank, and with every seed, the mycorrhiza is laid, a pellet of mycorrhiza is laid beneath the seed. As well, we can seed coat it, so use it for seed dressing, preferably when there are no fungicides in the seed dressing. And as well, and this is the next example, we can mix it with the seed, for example, with a cover crop seed. And you see that here in the picture in the right corner, there is the seed, and the bigger particles, those pellets, this is the mycorrhiza. And this is a very elegant method to introduce mycorrhiza into your system, into your crop rotation. Let's say if you do cover crop, mix it with the mycorrhiza, and when the main crop, the cash crop is coming, in this case corn, it will profit of this symbiosis. And in this case, we had a yield increase of this silage maize of over 20%. This is a lot, and I won't promise that this will be the case always. But of course, we are very happy to see such results under field conditions, which is the most challenging conditions we have. It's much easier to work in greenhouse, under greenhouse conditions. What you can see as well is that these plant pictures of the roots, the left side is untreated, the right side is of the inoculated plants, and you see that the fine root development is much stronger with the mycorrhiza. This is a classical result. We often have that the fine roots are stronger with the mycorrhiza. We have strategies to for the future, let's say, how the symbiosis can become more effective. You can work with the fungus to, let's say, train it, for example, that it works in a situation where there is quite a lot of phosphorus in the soil. this is what we do. And as well, there are groups working on the plant side. So they breed the plant in the direction that they are more open to the symbiosis, even under high P contents. And what we do as well, and we have a patent on this, is we use signal molecules to make the plant more collaborative, let's say, more open towards these fungi. Yeah, this is a summary of all the different factors which are affecting mycorrhiza, but as well all other microorganisms in the soil, and they are several. So it is really, as farmers, all farmers know, it's not easy to do farming. Every year is different. We have different climate, we have different culture varieties. And of course, the microbiome, this is what I am concentrating on in the soil, is different too. And then I have some more practical examples for you. This one is from commercial tomato production in Germany. It is done in this, in protected, because it's too cold in Germany. You have to do it in greenhouses, but it is done in these greenhouses. And we had quite a success, let's say, with this result. To summarize it, it is that tomatoes which have been grown with mycorrhiza are sweeter. So everybody called us after the publication of this. A lot of journals were interested in that. And I don't need to go into the details, but the tomatoes, and this is here the red bars, the red tomatoes, they are sweeter and they have more amino acids inside. So they are healthier, let's say. So this is an example where not necessarily we have more yield, but the quality is enhanced. And this is welcome, of course, too. And now I have some pictures. This is an example of roof greening in Europe. In some countries, there are laws that big roofs of supermarkets, for example, they have to be greened to keep the water there, to retain the water. And this water retention is not working well when there is a weak plant development. So on the left side, you see the situation without mycorrhiza, and on the right side with mycorrhiza. And you can easily see that with the mycorrhiza, the plants are much more happy, much more growing, more plants germinated, and the plants are bigger. And this is then an advantage. This is more or less a similar direction. But this is not made by seeds, but with pre-cultivated mats of these plants. So they are produced in the field and then cut off the field and brought to the roofs and unrolled there. And you can easily see this left side of this red line, there is untreated, and the right side is much greener, much more plant coverage, let's say. And another example in that direction, this is the deposit of a tunnel construction site in Switzerland through the Gotthard mountain. They digged a tunnel, put all these masses of gravel in another village, and you see the railroad track there. There was a heavy rain and all the gravel eroded. The railroad track was closed, had to be closed. So people were very unhappy, and we were asked to make a demonstration site there that with mycorrhiza, the plants are able to grow there. Without, it looks like on the moon, and the mycorrhiza sticks together these gravel particles, and so this material can stay there. It's nothing where you can yield something or somebody would be interested. It's just erosion control, but important tool. Yeah, this is an example of onion. So I just picked some examples because I didn't know what exactly you grow. So I have different examples, and to see that the application of mycorrhiza is quite broadly possible, let's say. That was seed coating. So these onion seeds were dressed, and you see both in the graph and in the picture that the treated shoot fresh weight increased a lot. So with the treatment, the shoot fresh weight increased a lot. that much that you can hardly believe it. The plants are really much bigger. But this was not only our product which was tested here, but I only put the picture of our product. So other products worked well too. So the situation was that these untreated control plants, they were really waiting for something to help them. I guess, I don't know, I don't remember the details. We didn't do this on our own. But there must have been really scarce nutrients in the soil. And then you have such results. Of course, everybody's happy to see that. Another series of examples from ornamentals, and these are 9 different ornamental species out of a nursery in Germany. And as well, you see that mycorrhiza is enhancing the root growth. In many of the pictures, you can see just with your eyes, some are a bit dark, but in most of them, you can easily see that there is more root growth with AMF than without. And yeah, I think that is the summary now. So this would be my take-home message for you as a concept for a reasonable use of mycorrhiza fungi in green business. So not only in agriculture, but everywhere. Some are rather trivial, I would say. So I would be skeptical with miracle cure because, yeah, if everything would be so easy and so perfect, everybody would use it. But those results which I have shown, I don't want to make them small, but if companies promise the moon, I wouldn't believe it. Use rich and active inoculum. And this, of course, you cannot see, but you can ask. You can ask how many propagules, which are the species in your inoculum, how many mycorrhizal propagules are inside, maybe how did you produce it in the greenhouse or not, and if there is no answers, I would be careful. Then apply inoculum cost efficiently, so don't spray it everywhere, spread it everywhere, but really take care that it comes near to the roots, that it doesn't lay on top soil, on the top of the soil in the sun, and is because the sun is sterilizing the mycorrhiza, or all other microorganisms too. The last in this row is don't forget ecosystem services as positive effects. So with that, I mean this kind of soil aggregation and water retention in the soil, because this is a very important step. Not very easy to measure, but possible. And then I had to cut a bit because we didn't have, we don't have the whole evening now. I didn't want to make it too long. Those modern approaches, I showed the tomato example. So mycorrhiza can not only enhance yield, but as well quality. Then there are modern strategies, let's say, how we acclimatize our fungi to the environment. And as well, it is possible and necessary to manage the natural AMF community. So people who treat their soil with care over time, and when you do this 5, 10, 15 years, mycorrhiza is coming back. If you want to speed up that process a bit, introduce mycorrhiza into the system. Yeah, I think that was all. On the very last slide, there is part of the team. My thanks to the PhD students who did the work partly, and my email address if you need it. And now I think we have time for questions. Yeah, it's a quarter of an hour, 3/4 of an hour, so that was good. I will stop the presentation because then I can see you again. And please feel free to ask questions. Let me check if there are some in the chat. I don't see questions in the chat. So maybe feel free just to open your microphone.
Hello, doctor.
Hi, Bruce. Hello.
My name's Bruce. I've just got 2 questions.
Yes,
What would be your #1 tip for improving your soil health sort of long term? And if you have a lot of manure, would you apply it directly to your fields for field health in terms of growing either a maize crop or a sugarcane crop? or would you compost it? What would be your advice there for long-term benefit? Thank you.
So the one question is if you would apply the manure directly to the field or compost it. Is that right?
Yes, that's one. And then what is your number one tip for long?
I am talking already for days, so I can only remember one question at a time. Yeah, thanks for the question. Well, for the mycorrhizal fungi, they don't mind, I would say. So there's no big difference. The deviation via the compost adds some more microorganisms to the stuff, let's say. So in general, compost is a very precious amendment to the soil. And then this addition of compost, that will enhance soil life in general over years. But it won't bring mycorrhiza into the system. And what again was the first question? Sorry.
What would be your number one tip on improving your soil health long term?
Yeah, that would be cover crop. A diverse mixture of cover crop, because that brings so many different advantages, from soil life to insects to so this is really organic matter, and it is rather easy to establish. Of course, as everything, we have to find or try which mixture of plants is suitable for your location, your soils, and I know people do it like that. Yeah, that would be the simple answer. I would start with that. Of course, reducing the pesticide treatments is always a good idea if possible. I would expect, and this is what people see, that once they use these methods, cover crop and others, then they can go down with their pesticide spraying. So it is a positive circle. Once you start it, it will get better and better.
Thank you very much.
You're welcome. There's another hand. Please.
Yes.
Doctor, I don't know if I'm interrupting anybody.
Yeah, sorry, my hand was up. I'll just quickly ask you, Doctor, a few questions. Of the 300 species, how do we know which is the correct one for Zambia?
Yeah, this is what we do. So we selected 3 or 5 out of those, and we do this work. So we select generalists, which work with a broad range of plant species, and they are not at all, these 300, they are not at all able to grow fast enough. It was, the number is just that they occur in nature. So this is why I mentioned that.
So the species that could work in Germany, it could also work in Zambia.
Yes.
It's not specific to the country.
No, on the species level not, but still we are a bit careful in spreading microorganisms all over the world. I know that there are European products here on the market from Spain, for example. Apparently they are working well, and I would always, maybe they did that check a bit to get a bit of feeling and experience of what species are here naturally occurring in different soils. And I just got today here from the farm soil samples to check and get a first information on how many different species are there under different managed crops, and how much mycorrhiza is there. So it's just that the keyword is invasive species. So I won't I wouldn't like the idea that I am the one waking up in the morning and am in the newspaper because one of my fungi did something, which is not expected at all, but I am always a bit careful, let's say, to spread it everywhere.
Okay, so the other thing, Doctor, is we've been told by some people that if your phosphate levels are above 25 parts per million, then the plant doesn't extrude the sugars to attract the mycorrhiza, and that you're wasting your money. But I don't know, there seems to be a conflict on that.
It is. 25 is a bit low, so to our experience, it starts with 100 ppm, which is 4 times more. But for sure, that is a challenge that the plants know when there is enough P in the soil, they don't have to pay the fungus for the service. But which did not work perfectly in evolution is that when the plant doesn't let the fungus in, the plant and the soil and the farmer cannot take advantage of all the other beneficial effects. So we are working towards fungi, and we do this via training of the fungi that work under these high P conditions. And at the same time, you can work on the plant side. So breed the plant, cut this kind of regulation mechanism a bit or depress it a bit. There's a group in Munich working on that, breeding plants, that the system is working even under high P conditions. Although I would say, yeah, at least in Europe, this will go down anyway, because P is not very cheap anymore. So when you check your soil samples and you have a high P, just stop using fertilizer. So it will go down.
Okay. And then the final question, sorry.
Yeah.
Okay, sorry, the final question, sorry to keep going, is there was also a concern of mixing Trichoderma asperellum with the Mycorrhiza, that the Trichoderma, that specific strain of Trichoderma is very virulent and aggressive and will eat the Mycorrhiza. I don't know what your experiences are, because sometimes we trying to mix on the seed the Trichoderma and the Mycorrhiza together. I don't know what your feelings are on that.
So I have only good experience with Trichoderma, but you know, there are many strains. Some are even plant protection products, so they are used as pesticide. We tried Mycorrhiza and Trichoderma together, and we didn't face any problem. So there was no, let's say, added value, but they didn't eat up each other. So depends, for sure, it depends on the Trichoderma strain. What does it do? But my experience is that we didn't have any problems.
Okay, thanks very much, and thanks very much for your lecture. We appreciate it very much. Thanks a lot.
You're welcome. Thank you. Tim is the next.
Good afternoon, Doctor. Can I just jump in with a quick question? What about carrots? Does it work on carrots?
Carrots, yes. Very easy.
Okay.
Yeah.
And thank you for your talk. Where can we buy your product?
Yeah, I don't know. In fact, we have a web shop, but I am mainly here, let's say, not purely for business, but for sharing knowledge. So we are just thinking if we should exhibit on your Agro Expo in next April. And then maybe we have more details and yeah, if you are interested, maybe you just send me a mail, so that we have no distributor here in your country.
Doctor, thank you for your time. Someone else can ask now.
Thank you. Ben is the next.
Hi, Doctor. I'm growing coffee in Zambia, and I'm very keen in introducing more mycorrhiza into it. But it's got a very specific season. You know, we're dry for 8 months of the year, it rains for 4 months of the year. And then during the rains, you know, we pour on a lot of synthetic fertilizer. So I'm just wondering when's the best time to add it, and which products would work best, because obviously it's a tree crop.
Yeah, it needs arbuscular mycorrhiza. Coffee is a host for arbuscular mycorrhiza, so this is easy. And I would apply it either when you plant the plants, or when they are very young, when you have them in the nursery, because then you only need a small amount and it makes the application cheaper. If the plantation is already there, you can add it to the soil, so bring it to the roots. Take care that it's not laying on top of the soil or any mulch or so, because otherwise the sun will kill it. And a dry season is no problem. The product itself is a very, very dry powder. And when it lays in the soil and it is dry, it just won't grow, but it won't die as well. So the spores are used to that, so they can stand dry periods. And so shouldn't be a problem that of these long dry periods, let's say. But they won't grow during that time, as the roots maybe they will just wait until the water is coming.
So you're not concerned about them being suppressed by all of the fertilizer that I will put on?
This problem everybody has. So when it comes to very high P fertilization, and you cannot change it or don't want to change it, or you, of course, I know this is the system how it is or was, but why not try it? Go down with the P supplement a bit, not everywhere, but in like a trial field, and add mycorrhiza to that. So it's worth trying.
And just lastly, I mean, you mentioned obviously if I apply when we plant the actual seed in the nursery, that would be best. I do have trees in the field, and you think a once-off application is fine, or you must repeat every year?
That depends a bit on your management or conditions. Depending how beneficial, let's say, or how careful you can be able to manage your soils. I know we all have forces, or we are all forced to work in a certain way. So sometimes it doesn't help. I cannot recommend to use mycorrhiza. For example, for crop rotation, where there's a lot of non-hosts in the crop rotation. So we have that in Germany. They use sugar beet, and then after canola, and in the meantime they have as a cover crop maybe mustard. And then I have to say, please save the money, don't use the mycorrhiza. So, but when people are able to change things and willing to change things, there's always a way. Just keep in mind the most, let's say, harmful factors are, of course, uncovered soil. This is not good. Repeated pesticide treatment for a long time. This is not the best for soil life in general. And very high mineral fertilizer application too. So those, if you will be able to adapt these factors a bit, I think you will come along.
Excellent. Thanks very much, Doctor. Really good presentation. Thanks for your help.
Thank you. It's good that you liked it. So there's another hand. Rassi, yeah.
Hi, Doctor. I'm using Rassi's computer, so it's Sally here. I'd like to know if we have good compost and we grow certain plants like corn, maize here, could we grow our own mycorrhizal fungi, or are there other species of plants that we could grow to make our own mycorrhizal fungi?
Okay. Yeah, your connection is very weak, but I think I got the question. For everybody, the question was if to enhance the mycorrhiza, is it the best to use corn, or can other species work as well, plant species? Corn is good, but other species work as well. So you can as well use grasses or clover, so this depends a bit on how big the plants can grow, or if the space is limited somehow. We do use a mixture of plants, so this is a good idea too, because they all have different root architecture. Some are going deep in the soil, some not. Some are very fine, some not. So I would use a mixture of plants. And what we do as well, change from time to time. So introduce a new species into the production, leave away one for 1 or 2 years. So keep the fungus a bit active with this kind of challenges, because otherwise they can get lazy. This is a problem for this lab production method too. those fungi, they are supplied with everything what they need, sugar everywhere, and in the end they are not that keen anymore on discussing and communicate with plants, because they are lazy due to the production. And for the production with different plant species, it's a bit the same. If we would only use corn and again corn, the population of the fungus will be narrowed a bit through that. So I would recommend a mixture.
Sorry, so we have to buy mycorrhizal, we cannot make our own.
I couldn't understand. You bought
Can we
mycorrhiza?
grow our own from taking a bit of forest soil and putting it in a biocomplete compost? And then we grow plants? Can we make our own?
If I only understood some words, but if the question was if you can grow your own mycorrhiza, yes, this is possible. But of course, if you are not able to look through the microscope to stain the roots, of course, everybody is able to learn that, but you have to go somewhere, then you cannot control it. But it is possible. And if you have with your method, if you have good experiences, this is something. I would do it, try it, and see what happens. But if you, of course, you don't know which fungi are inside and how much. This is the gap.
Yeah. Okay. Thank you very much.
Thank you. Aha, now I see questions in the chat. Let me have a look. Yeah, that was the coffee question. Simon has a question. You said if plant is happy, it won't make an association. But why? Surely it's an interest to hook up with mycorrhiza no matter what. Yeah, I think that was now already explained, that because the plant is working economically, and as they have to pay with sugar, when they are happy, they don't spend extra sugar money. So that's the explanation, but I think that was already old, let's say, the question. Yeah, I'm happy that you had questions, interesting questions, but apparently I was able to answer them more or less. How to go on? If there are no other questions, I might hand over again to, yeah. Okay, so thank you, bye-bye. Thanks for having me here in Zambia. I liked it a lot. And ask me via email if more questions occur. So I will hand over now.
Thank you very much.
Yeah, thank you very much for joining. This was the first time that we had a Zoom meeting like that. So we faced some hiccups, but we also see the potential now, and hopefully in future we can also get experts maybe from Germany, and we just do an online meeting like that. I'm happy if you give me ideas in what you are interested in, and we try to find something or some experts who could advise you and who can answer your questions. As Dr. Schneider pointed out, all these mycorrhiza, cover crop, and we all know it has a positive effect, but nobody really knows all the answers to all the questions. So there's still a lot of trial and error, and it's nice to have a good exchange between the farmers. We also had very nice discussions in the field yesterday with the cover crop we grew and so on. If you come if you pass AKTC, please pop in, have a coffee with us, and we can chat. Otherwise, thank you very much for attending, and we will share the link to the recorded presentation so that others can watch it as well. Thank you very much, and enjoy your evening.
Thank you very much, Helmut. We appreciate it. Thanks very much.
Cheers.